กรมศรีสวัสดิการจังหวัดน่าน School. Today we are going to present about the study and comparison of the effectiveness of wet and dry termite-nest microorganisms on the decomposition of various types of agricultural material and the effect on gas production and NPK values.
In many communities, regular cutting of tree, branches and grass generate large amount of plant waste, which is often disposed of by open burning, causing smoke and air pollution. At the same time, termites cause significant damage to wooden structure, and chemical control methods may pose risk to human health and the environment. Therefore, this project integrates local wisdom related to termite with scientific principles to investigate effective fermentation formulations for producing bioextracts that can be used to decompose agricultural residue and to spray on homegrown vegetables, promoting chemical-free cultivation and environmental sustainability. Objective: first, to develop termite-nest microorganisms using different raw material in both wet and dry formulation evaluated by gas production and NPK value. Second, to compare the composition efficiency of agricultural materials using its formulation of termite-nest microorganisms.
Experimental method, Part 1. To study and identify production formulas for termite-nest microorganisms in both wet and dry fermentation using different types of materials. First, termite-nest 40 grams was mixed with each material 200 grams, such as raw rice, molasses, sticky rice, cooked rice, raw sugar, and pond water 600 ml. Placed in a soda bottle equipped with a pressure gauge to measure gas production. Second, the wet fermentation was carried out for 7 days. Third, for dry fermentation, no water was added initially. The mixture was left for 3 days. Then water was added on day 4, and fermentation continued until day 7. Fourth, the amount of gas produced, NPK value, and pH.
The result from Part 1 showed that first, termite-nest microorganisms could be successfully produced in various formulations: chemical reaction in the fermentation process, hydrolysis and fermentation of starches and sugars. Starch is enzymatically hydrolyzed by amylase into simple sugar, as shown in the equation. Monosaccharides are fermented by microorganisms in anaerobic conditions, as shown in the equation, and producing organic acids such as lactic acid, as shown in the equation. Second, the material had the highest gas production was molasses, followed by sticky rice, cooked rice, brown sugar, and raw rice. The production of carbon dioxide gas indicates microbial activity. Molasses serve as a source of small, readily soluble sugar molecules and already contains mineral, allowing microorganisms to utilize it more rapidly than cooked rice and sticky rice, which must first undergo stress degradation despite being cooked stretch. Brown sugar is a simple sugar. It contains fewer supplementary nutrients. Therefore, microbial growth is limited and gas production is lower. Third, the dye formula, or the formula that we left for 3 days before adding water, produced higher NPK values than the wet formula. This is because dry fermentation allows termite-nest microorganisms to proliferate without dilution. Although gas production is relatively low during the initial stage, the subsequent addition of water-released nutrients accumulate with microbial cells, leading to higher NPK value compared with the wet fermentation method. Fourth, the termite-nest microorganisms made from molasses show higher NPK values than those made from raw rice, sticky rice, cooked rice, and brown sugar. This is because molasses is a source of small molecule sugar that microorganisms can readily utilize, and it naturally contains mineral, particularly potassium, which promote microbial growth, gas production, and nutrients accumulation at a high level.
Part 2. To compare the decomposition efficiency of agricultural materials across different formulation of termite-nest microorganisms. First, 6 strands of rice store, each 15 cm long, and 2 pieces of red tea leaf, with a total mass of 1 gram, were soaked in 10% volume by volume fermented solution 100 ml of 14 days. Second, then test for micronization. The degree of rice store decomposition was tested using a weight learning method. In rice store, strands that broke easily indicate a higher level for decomposition. The amount of weight loss was measured after decomposition occurred.
The result from Part 2 showed that first, efficiency of termite-nest microorganisms in decomposing agricultural material, and second, the termite-nest microorganisms made from raw rice decompose leaf more effectively than those made from molasses, sticky rice, cooked rice, and brown sugar. This is because raw rice releases high carbohydrate salts gradually, resulting in more stable fermentation system, a slower decrease in pH, and favorable conditions for the continuous activity of plant cell wall degrading enzymes, such as cellulase and hemicellulase. In contrast, rapidly fermenting formulation cause acid accumulation that may inhibit long-term enzyme activity, which may inhibit enzyme activity over time and consequently reduce their efficiency of leaf decomposition compared with the raw rice formulation.
In this experiment, EM was used for decomposition, and the results were compared with those of termite-nest microorganisms. It was found that termite-nest microorganisms decompose leaf more effectively than EM because it contains microorganisms that naturally break down leaf and plant fiber As a result, the enzyme remain active longer, leading to greater leaf softening and decomposition. Their study found that termite-nest microorganisms can be used to decompose plant material and develop into the powder form that we make from raw rice for easier storage and application.
Utilization. First, channelage with committee on wet and dry fermentation. Second, recommend practical application for agricultural waste decomposition and plant foliar nutrient. Third, develop into a powdered form for easier storage and application.
Fourth, produce a cell wet and powdered formulation at affordable price via online and on-site. Fifth, promote chemical-free, vegetable consumption and environmentally friendly waste management.
Convert termites complex into beneficial agent for natural ecosystem restoration.
Thank you for your attention.